Biorefinery systems engineering, bioethanol generation and co-product optimization from food waste. The ever-growing global population creates a significant challenge for the food waste. Canteen food waste, comprising one-third of the global food production, poses a significant environmental burden due to methane emissions for overflowing landfills. Creating a sustainable and integrated biorefinery process to convert diverse food waste into valuable byproducts like bioethanol and bio-based materials. Objectives: Bioethanol: unlock sugars from food waste to create this clean burning fuel. Biocapsules: evaluate biofilms for potential use as medical capsules. Biopolymers: explore using lignin and butterfly pea flour. Work plan: food waste processing, feedstock characterization, fractionation, component analysis, bioethanol production, and lignin valorization.
Includes food waste pre-processing. In the first step, food waste collection. We have collected cauliflower leaves, banana peels, beetroot peel, potato peel and orange peel. Then we need to chop them into fine pieces. Then we need to add the nutrients to adjust the pH. We have sent the feedstock to the Usmania University for the GCMS analysis. This is the result we got. Alkaline pre-treatment. In this we have to prepare 5 test tubes, each labeled with the corresponding NaOH concentration. First we need to add the prepared NaOH solution. Repeat with the HCl and leave it for 24 hours to stir the mixture. Step is cellulose and hemicellulose extraction. After incubation, carefully filter the content by using filter paper, mainly the lignin. Once the cellulose is precipitated, separate it from the filtration. Add a small amount of sulfuric acid. Add isopropanol to the remaining solution containing the solubilized hemicellulose. Lignin quantification: the solid residue remaining on the filter paper after the initial filtration, it represents lignin. Then weigh the dried lignin residue and calculate its mass as a percentage of the initial food waste. This provides an estimation of the lignin content in the food waste. To calculate the cellulose, hemicellulose and lignin, the amount of cellulose, hemicellulose and lignin after calculation.
FTIR analysis. By comparing our sample with the reference sample, we found that our sample consists of cellulose, hemicellulose and lignin. Thermogravimetric analysis. Our sample is compared with the ideal sample and it is found to be that similar.
For further fermentation, we have to prepare fermentation media. Then we need to autoclave the fermentation solution at 121 degrees Celsius under the pressure for 15 to 30 minutes. Then we need to cool the solution at room temperature. We need to add yeast of about 10 grams to convert the sugars to ethanol. We need to place the solution at shaking incubator. Then measure the absorbance by using spectrophotometer at 600 nanometers. In hydrolysis, we have used acid alone. 1% of H2SO4, that means 10 ml of H2SO4 dissolved in 1000 ml of distilled water. And the last step is distillation and dehydration. First, we need to heat the fermentation mixture solution obtained from the food waste into the distillation mixture. Boiling point of ethanol is lower than the water, so ethanol vaporizes first. In the receiving flask, we have to collect the distillate, which is known as crude ethanol. To remove the water, we have to use dehydration process. In biopolymer formation, separation of petals from butterfly pea flowers, boil the flower petals, filtration, addition of cornstarch, then the blue polymer is formed. Biofilm formation. At first, we have to add 1.15 grams of CMC. It acts as a base material. Then 0.15 grams of citric acid acts as a binding agent. We have to place the solution on a magnetic stirrer for about 1 hour. Then pour the solution on your petri plates. Let it cool.
To calculate the amount of bioethanol produced from food waste, we have took the standard plot of ethanol. From the graph, we have took the values and we have found that we have produced 207 ml of bioethanol for 70 grams of food waste, and where a graph has been drawn for the observed readings. Qualitative analysis, where it was done by Jones oxidation reaction, where we took 2% of potassium diachromate solution. The change in color from orange to blue states that the ethanol was converted into acetic acid. The SEM analysis gave us a clear idea of the structure at its microscopic level. To calculate the amount of anthocyanin present in the biopolymer, we took a test tube and added 2 to 3 grams of biopolymer to it and added it to a sodium acetate buffer which is of pH 4.5, and we also made a blank solution with no biopolymer, only the sodium acetate buffer. Using the formula, we have calculated the amount of anthocyanin content in the biopolymer and it was found to be 33.7 mg per g. For testing biofilm, we did few experiments like swelling ratio and degradation test. The percentage of degradation was identified using degradation test. Every 3 days, the weight of the sample biofilm was measured. The table shows the amount of biodegradation. Future prospects of the project are wound dressing, tissue engineering, samples, and biocompatibility and biodegradability tests. This step process successfully transforms the canteen food waste into valuable bioproducts, including bioethanol and bio-based materials, showcasing innovative waste management and biorefinery technology.